in the textbook, if I remember correctly, I think that's where I read this. This part of the brain is sometimes called the gateway to the cerebrum, because almost all sensory pathways travel to this part of the brain. Okay, I think most of you have answered, so let's take a look at the results. Most frequent answer was thalamus, and that's correct. Nearly all sensory pathways travel up the spinal cord through the brainstem to the thalamus. In the thalamus, those sensory pathways synapse with third-order neurons that take it up to the primary somatosensory cortex, which is on the postcentral gyrus. That's where those sensory signals are interpreted. The only exception to that is the olfactory pathway, because that goes straight up and synapses on the olfactory bulbs of the frontal lobes. Okay, so today we're going to talk about senses and sensory receptors and sense organs and sensory pathways. So a sensory receptor is any structure that's specialized to detect a stimulus. And sensory receptors are divided up into two types. They're the general senses, which are rather simple in structure, and the special senses, which are found in sense organs that are a lot more complex in structure. So, some of the special senses, such as the sense of vision, the sense of olfaction, which is smelling, the sense of gustation, which is taste, those sensory receptors are actually modified epithelial cells. So they're not made of it's not a neural cell. Okay? It's a modified epithelial cell. So those sensory receptors respond by releasing a neurotransmitter onto another cell, which is a neuron. That neuron then can produce an action potential. Now, there is one of the senses, though, that is not a modified epithelial cell, and that is the olfactory receptors, which we're going to talk about today. Olfactory receptors actually is a neuron with an axon. So it is able to generate action potentials. Those action potentials travel up the axon of the olfactory receptors through the cribriform plate, which is in the floor of the cranium, and then it synapses right on the olfactory bulbs. So that is actually a neuron. Now, other general senses, like the sense of pain, the sense of cold, sense of pain and cold are basically unmodified sensory neurons. So the part that responds to the stimulus is just the dendrites of a sensory neuron. So some of them are just simply unmodified sensory neurons. There are other general senses, such as the sense of touch, which consists of a sensory neuron that has a non-neural tissue surrounding the dendrites. This non-neural tissue is usually just a layer of one or more layers, actually, of connective tissue surrounding the dendrites. Okay, those types of receptors form some of the touch receptors, of which there are more than one type, as well as pressure receptors. So they're just a sensory neuron with connective tissue surrounding the dendrites. Sense organs, as we've seen in lab, can be much more complex. They're made up of a number of different tissue types to form a true organ. So the eye is actually a sense organ, as well as the ear, and the taste receptors, which are located in taste buds. Taste bud also forms an organ. Now, no matter what the type of sensory receptors, all sensory receptors function as a transducer. A transducer is any device that converts one form of energy into a different form of energy. So there are man-made transducers that are used in everyday life, along with natural transducers that act as sensory receptors in the body. So for an example, one example of an artificial transducer that actually simulates the function of the ear is a cochlear implant. Cochlear implant is an electronic device that's implanted in the inner ear of people that are deaf, sometimes people that are born deaf, that are not able to hear. So what this cochlear implant does is it converts sound waves, which are kind of like vibrations of air molecules, converts that energy into electrical impulses that stimulate a cochlear branch of the vestibulocochlear nerve. And that allows deaf people to hear, sometimes for the first time. Okay? It's the same way that the cochlea works in the ear of most people that can hear. The cochlea converts sound waves into neural impulses, which is a type of electrical form of energy because it involves the flow of ions, which is a current. So other sensory receptors convert other forms of energy like light or heat or touch or vibration or pressure into a different form of energy, which makes up the neural signals. All right, now, sensory receptors work much in the same way as an ordinary neuron. So when an ordinary neuron is stimulated on its dendrites, that produces a graded or local potential, which doesn't travel very far. It just travels down the dendrites over the cell body to the first part of the axon. And that's similar to the way that most neural sensory receptors work, except in a sensory receptor, when that local potential is produced by a stimulus, that local potential is called a receptor potential. So receptor potential is just a local potential that's produced on a sensory receptor. Okay? So if that receptor potential then is above a threshold, when it gets to the axon hillock, then it will trigger an action potential, which sends a signal up towards the brain that allows the brain to detect that stimulus. Now, the sensory receptors, though, that are modified epithelial cells, like the photoreceptors and the taste receptors, and also the hair cells, those respond to a stimulus not by producing receptor potential, but by releasing. They respond not by eventually producing action potential, but they respond by releasing neurotransmitters. So those modified epithelial cells work by a slightly different mechanism. All right, now, during every day, we are constantly being bombarded by sensory stimuli. But not all of those produce a neural impulse that is detected by the brain. a lot of that information is filtered out in the brainstem before reaching the sensory cortex. And so that prevents the brain from being overloaded by sensory stimuli. So only sensory signals that are large enough or that are deemed to be important reach the brain. And if a signal does reach the sensory cortex and it allows us to be aware of that stimulus, then that's called a sensation. Okay? So sensation is when the brain receives a stimulus and we're aware that the brain has been stimulated. Okay? Now, a perception is different than a sensation. Sensation is just the arrival of a signal at the brain, and it alerts the brain that something has, that we've been exposed to a stimulus. A perception only occurs when a sensation is interpreted and categorized. So in order for perception to occur, that sensory information has to be processed. It has to be processed by the primary somatosensory cortex, and that information has to be integrated with stored information in association areas of the brain. So there's a considerable amount of processing that has to occur before perception occurs. A perception then allows us to determine the type of stimulus, or what's called the modality of stimulus. So it allows us to determine that if that stimulus was light touch, or whether it's heat, or whether it's pressure, or whether it's light. Okay? A sensation just alerts our brain that we've been exposed to something, but you don't necessarily know what it is. Okay? It's kind of like if you're kind of drowsy and falling off to sleep, and something that wakes you up and you're not quite sure what it is, that'd be a sensation. If you can tell what kind of stimulus it is, and if you can tell more information about it, like the type of stimulus, how intense it is, the location of the stimulus, then that becomes a perception. So it is possible to have a sensation without a perception. You cannot have a perception, however, unless you first have a sensation. All right, now, there's different kinds of information that's important for the brain to be aware of about any one sensory stimulus. It's important in order to maintain homeostasis for our brain to know the type of stimulus, which is called the modality. It's also important for our brain to know what part of our body was stimulated. Okay? That's the location of the stimulus on the body. It's also important for our brain to know the intensity of the stimulus, because if it's a very low intensity stimulus, it may not be important enough to respond to. But if it's a very large magnitude stimulus, it may be important. So the brain needs to know something about the intensity and the duration. The brain needs to know how long that stimulus is being applied, because if it's only applied for a split second, it may not be that big of a deal. If you apply heat for a long time, though, that may become a big deal. So that's kind of why, if you just run your hand through the flame of a candle very quickly, it's not going to burn you. It's short duration. It's not that important. But if you hold your hand over that flame for a long time, that longer duration stimulus is going to be important. Now, the perplexing thing about all of these types of information is that all action potentials which carry those signals to the brain are the same. If all action potentials are the same, then how does the brain know that an action potential was produced by the eye, or was it produced by the ear? Okay, that's modality. Given that all action potentials are the same. Well, there's different ways that the nervous system encodes these four different types of information, okay, using action potentials, which are basically all the same. So the way in which the nervous system encodes the stimulus modality, or the type of stimulus, is by having separate pathways from each of the receptors that are able to detect the type of stimulus. So every stimulus has a different kind of receptor that's most responsive to that stimulus. And that type of receptor has a separate pathway to a unique part of the brain that interprets that stimulus. So this form of encoding is called the labeled line code of stimulus modality, because every different modality of stimulus has a separate pathway or a separate line to the brain. And that way the brain can distinguish that type of stimulus from other stimuli, because it has a separate pathway so that your signals don't get crossed or mixed up, and it travels to a different part of the brain that's responsible for interpreting that type of stimulus. Okay? So this is a diagram that shows how this works. So for example, the taste receptors on the tongue, those are the only type of receptors that can detect the different types of taste that our tongue is able to detect. And so different tastes are detected on the tongue by taste receptors. Those signals take a unique pathway to the part of the brain that interprets that sense of taste. And so that allows the brain to know that that's a sense of taste, as opposed to a sense of hearing. Because hearing has a different pathway that takes to a different part of the brain. So it's almost like the brain is made up of a large number of different processing centers, each of which processes just a single type of stimulus. Okay, now, it's also important that the brain be able to determine what part of our body was stimulus, or the stimulus location. The way that the brain determines the stimulus location is similar to the way that it detects different modalities. There are unique pathways from each different part of the body to a unique part of the brain that's responsible for interpreting signals from that area of the body. Okay, you remember that that map of the primary somatosensory cortex that had the body parts arranged along it? That's called a homunculus map. Those body parts indicate what part of that gyrus is responsible for interpreting signals from different parts of the body. So, for example, sensory stimuli from your hand travels up your arm to your spinal cord, and then up the spinal cord to the thalamus. And then from there, those signals are carried to the part of the primary somatosensory cortex that is responsible for interpreting signals from your hand. So it's a unique pathway to a unique area of the brain that's responsible for that location of the body. So in your hand, there's an area surrounding each sensory receptor that, when stimulated, will stimulate that receptor. Okay? For example, let's say you have a sensory receptor on your hand right here. Around every receptor, there's an area, it's usually a circular area, called a receptive field. And if any part of that receptive field is stimulated, that will stimulate that receptor, and that signal then travels to the part of the primary somatosensory cortex that's responsible for detecting stimuli from this receptive field. And the number and size of receptive fields on your skin differs depending on the part of the body. The skin along your trunk, for example, doesn't have very many sensory receptors. And so the receptive fields on your trunk are very large. And so that means it's a little bit harder to detect the exact point at which an area of your trunk is being stimulated. Your hand and your fingertips, though, have a lot more receptive fields because they have a lot more receptors. And so you can pinpoint sensations on your hand and fingers a lot better than you can on your trunk because there's more sensory receptors, more receptive fields. Receptive fields are smaller because they're packed more tightly together. And so you can localize touch a lot better on your fingertips. So this is called the labeled line code of stimulus location because each sensory receptor in each part of the body has its own separate pathway to the part of the brain that interprets that signal. So this is a diagram showing that post-central gyrus, where the primary somatosensory cortex is located. It's that ridge just posterior to the central, or just posterior to the central sulcus. And you can see on the right, the body parts indicate what parts of that gyrus are responsible for interpreting signals from the different areas of the body. So that's called the labeled line code of stimulus location. Now, stimulus intensity, on the other hand, is coded differently by the nervous system. There are two ways in which stimulus intensity is coded. One of them is according to the frequency of action potentials that are produced by the sensory receptor. So usually if a sensory receptor is stimulated, it doesn't result in just one action potential being produced. Usually it causes a whole burst of action potentials. And the frequency of those action potentials indicates to the brain the intensity of the stimulus. So a very large intensity stimulus produces a very high frequency of action potentials. In other words, there's more action potentials that occur per second. A very weak stimulus, however, will only produce a very low frequency of action potentials. So you'll only have a few action potentials per second. So that's called a frequency code. So intensity is indicated by frequency. Also, another way that the brain can determine the intensity of the stimulus is according to the number of sensory receptors that are stimulated. Because usually a large stimulus stimulates more receptors than a small stimulus. And so the more receptors that get stimulated and send signals to the brain, the larger the stimulus. But it's the frequency that indicates the intensity of the stimulus. Now, stimulus duration, I think it's easy to understand how that works. The longer that burst of action potentials occurs, the brain is able to determine the duration according to the duration of the burst of action potentials that's produced. But stimulus duration occurs in two ways. These two ways in which stimulus duration occur, there are some sensory receptors that are called phasic and some that are called tonic. Let's start with tonic receptors first. Tonic receptors are sensory receptors that will continue to produce a burst of action potentials at the same frequency for as long as that stimulus is applied to the body. So one example of a tonic receptor are pain receptors. If a pain receptor is stimulated by a painful stimulus, that pain receptor will continue to respond for as long as that painful stimulus is being applied. And so the intensity of the action potentials produced by that pain receptor will not decrease as long as that painful stimulus is being applied. Okay. Now that's actually adaptive. Generally, we don't think that pain's a good thing, but pain actually has a purpose. Pain lets our brain know that there's something wrong, that there's something that is happening to our body that's damaging it. And if pain receptors didn't continue to respond while a painful stimulus was being applied, then our body could end up being damaged without our brain knowing it, and our brain wouldn't be able to respond to it. So pain receptors, also hair cells in our ear, those are also tonic. Hair cells continue to respond for as long as sound is bombarding our ears. Now other receptors though, like pressure receptors that are in our skin. Pressure receptors are a good example of a phasic receptor. Phasic receptor responds strongly at first to a stimulus, but then the response diminishes over time. Okay. Another example of a phasic receptor are the olfactory receptors. You've probably noticed this before. If you're sitting in a room and somebody walks into the room who has just sprayed themselves all over with perfume, when they first go in the room, you really notice that perfume. It smells very strong. But if they stay in the room for a long time, then after a while you don't notice it as much. Okay? After a while you may not even smell it all. Okay? Another example of a phasic receptor are temperature receptors, especially cold. Well, both hot and cold receptors are phasic. And hot and cold receptors are actually just naked nerve endings of sensory neurons. And cold, let me give you an example of how a cold receptor is phasic. At the beginning of a summer, if a swimming pool first opens, it's not really hot out yet, or it hasn't been hot for very long. The water in the swimming pool may still be pretty cold because it hasn't had a chance to warm up much. So if you jump in that swimming pool and the water's cold, you're really going to notice it. You know, it's going to feel really cold. In fact, you may gasp for air. It's so cold. So it's going to feel real cold at first. But if you stay in that water, for let's say 10 minutes. After 10 minutes, that water's not going to seem as cold anymore. You're not going to notice that it's so cold. It's going to feel more comfortable, actually. And that's because cold receptors are phasic. They respond first very strongly to a change in temperature. But then over time, their response diminishes. Hot receptors are the same way. If you jump in a hot tub that's 103 degrees, it's going to seem real hot at first, but after you've been in there for a while, it doesn't seem quite as hot. So tonic receptors include sense of olfaction, or not olfaction, that's phasic. Tonic receptors include pain receptors. Photoreceptors are somewhat tonic. Generally, as long as light reaches your eye, you're able to see. You don't go blind after a while because the photoreceptors no longer respond. They're mainly tonic, but they're somewhat phasic. in the hair cells in your ear and in the vestibular apparatus, those are also tonic. All right, any questions? These are kind of the two extremes, though. There are some senses that are kind of in between, but usually they're closer to one than the other. So they're mainly tonic or mainly basic. All right, now different types of receptors can be classified in a couple of different ways, and I don't want to dwell on this too long because I want to get to the sense of taste and the sense of olfaction today. One way that sensory receptors are classified is according to the stimulus modality or the type. So receptors that respond to changes in temperature, either heat or cold, are called thermoreceptors. Receptors like the ones in the eye that responds to light are photoreceptors. Pain receptors are called nociceptors. Receptors that respond to chemicals like the olfactory receptors and the taste receptors, those are called chemoreceptors because they respond in response to the binding of chemicals to the sensory receptor. And receptors that respond to vibration, touch, pressure, stretch, or tension are called mechanoreceptors because those are mechanical types of stimuli. Another way that receptors are classified is according to the origin of the stimulus, whether it comes external to our body, or internal. Stimuli that reach our body from outside of our body are called exteroceptors. So like temperature receptors would be exteroreceptors. Internal stimuli like internal pain from visceral organs, those are called interoceptors. And sensory receptors that are located in joints, tendons, and muscles are called proprioceptors. And those are the sensory receptors that are responsible for some of the spinal reflexes, like the stretch reflex, and they also allow the brain to determine the position of body parts. And then, as I said at the beginning of class today, overall sensory receptors are classified as general and special. General senses or general sensory receptors tend to be very simple in structure and they're very widespread on the body. Special senses are more complex and they tend to be concentrated in the head. They're just located exclusively in the head. Special senses include vision, hearing, olfaction, and gustation, taste. Okay, so let's take a quick look at some of the different types of general receptors. This first one are just free nerve endings, or what's sometimes called naked nerve endings. They are responsible for detecting pain and temperature. Some of them detect pain, some of them detect temperature, but they're just naked nerve endings. These three that I have circled right here are all examples of tactile receptors. Tactile receptors detect light touch, provides the sense of touch. And there's different types of light touch receptors. There's one type called a tactile corpuscle. It's made up of a sensory nerve with a connective tissue covering. Just a single layer of connective tissue covering the dendrites. These are located in the skin. There's another type that's called a tactile disc. A tactile disc is made up of a non-neural cell called a tactile cell that releases neurotransmitter in response to light touch. That neurotransmitter then stimulates another cell that actually is a nerve that produces an action potential that travels to the brain. And then a third type of tactile receptor is a is a hair receptor. And hair receptors are made up of a dendritic ending of a neuron that wraps around the base of a hair follicle. When the hair is touched, that stimulates the dendrite of the hair receptor. Then there are some others, like these three right here, that are responsible for detecting heavier pressure. Most common one is called a lamellar corpuscle. These also used to be called Pacinian corpuscles. They have many layers of connective tissue around around a single dendrite of the neuron. So these type of receptors only have one dendrite attached to the cell body. And lamellar corpuscles are very phasic. They respond strongly at first pressure. If that pressure continues to be applied, they'll quit responding after a while. Then we have proprioceptors, the muscle spindles, which are stretch receptors in the muscles. This is responsible for the stretch reflex. And tendon organs, which is responsible for the tendon organ reflex. Okay, let's take a look then at the other two special senses that we haven't studied in lab. The first one is gustation, which is a fancy word for taste. Taste is caused by a sensory receptor that's located in an organ called a taste bud. Taste bud is shaped sort of like a garlic bulb. You know, when you first dig up garlic, the garlic bulb is made up of a cluster of garlic cloves that you can pull apart. That's kind of the way a taste bud is structured. And gustatory receptors or taste receptors are classified as chemoreceptors because it responds to chemicals. And the chemicals that stimulate the taste receptors or gustatory receptors are called tastins. And these tastins stimulate a taste bud, and the taste buds are actually located on elevated areas of the tongue that are called lingual papillae. Lingual means tongue. And papillae are elevated structures. You can actually see these with it. They're small, but you can see them with the naked eye. They're the little elevated areas of your tongue. Okay? There's a picture of some of them here. The lingual papillae come in different shapes. There's some of them that are shaped like this. These are called valate papillae. There are others that are a lot more narrow, kind of spike-like. Those are called filiform papillae. And the taste buds are located on those papillae, which they're just simply elevated structures on the tongue. Now, down here in the lower right is what we need to focus on. This is a diagram of a taste bud. This is the organ of taste. Taste buds contain three different kinds of cells. The most important one is a taste cell, which is a gustatory receptor. The taste cells have little hair-like appendages called taste hairs. And when certain chemicals bind to the taste hairs, that causes the taste cell to release neurotransmitter onto the dendrites of a neuron. And that neuron then produces an action potential that travels through either one of two nerves, depending on what part of the tongue it's on. Remember, there's two cranial nerves that carry signals concerning taste. Does anybody remember the name of either one of those? the cranial nerves that carry signals concerning taste. One of them is the facial nerve that carries signals concerning taste from the anterior 2/3 of the tongue. So if the taste bud is on the anterior 2/3 of the tongue, those signals would travel through the facial nerve to the brain. The other one is the glossopharyngeal. Glossopharyngeal carries signals concerning taste from the posterior 1/3 of the tongue. And that travels to a taste center on the brain. Okay, so that's the taste cell. That's what acts as the taste receptor. There's also supporting cells. These blue cells are supporting cells. The supporting cells just hold the taste bud all together and helps to support the taste cells. And then at the base of the taste bud are smaller cells called basal cells. Basal cells are actually stem cells. And these basal cells are able to divide to produce new taste cells if the taste cells get damaged. Okay, if the taste cells die, the basal cell can multiply and it can replace those taste cells. You've probably kind of experienced this before. If you drink something that's really hot and it burns your tongue, usually your tongue heals pretty quickly. And that's because if these taste cells get killed by real hot liquid, these basal cells divide and replace them pretty quickly. So this is a microscopic structure right here. That's located on these elevated structures like this. It's called a papilla. Okay. The papilla just puts the taste buds up closer to the surface of your tongue where your food would be located. Now, it used to be that when you looked at anatomy and physiology textbooks, they would show a map of a tongue showing where different kinds of tastes could be detected. So they thought, for example, that sweet tests were the taste receptors that detected sweet tastes, they thought were just right along the tip of the tongue. And they thought that the bitter receptors were located near the base of the tongue. However, physiologists now think that those different modalities of taste are not just restricted to certain areas. They're actually found throughout the tongue. Now, some of them may be concentrated in or places, but they're found everywhere. In fact, there's some of them that are even found on the inside of your cheeks. And it used to be that they thought there were only 4 different modalities of taste. And those were salty tastes, sweet tastes, sour tastes, and bitter tastes. But now they think that there's 5. They've discovered a 5th type of taste receptors. This 5th sense of taste is called umami, which when I first heard that word, I thought it was a joke. It turns out it's a Japanese word, okay? And umami tastes are savory tastes that are produced by certain amino acids. So a lot of comfort foods have a umami taste because they have has a lot of protein. Like chicken soup has proteins from chicken that's made up of certain amino acids like aspartic and glutamic acid. Salty tastes are produced by metal ions such as sodium, potassium, salts. Sweet tastes are produced by carbohydrates such as sugar. Sour tastes are produced by acids. And bitter tastes are produced by toxic substances such as alkaloids that tend to be not as good for you. Okay. So, but these are scattered all over the body. Now, we're about out of time, so I want to go on and talk about olfaction. So, olfaction refers to the sense of smell, and the chemicals that stimulate olfactory receptors are called odorants. And there are a lot more types of olfactory receptors than there are taste receptors. We only know of 5 different kinds of taste receptors. But most people have about 350 different types of olfactory receptors. And so by stimulating different populations of these olfactory receptors, most people can distinguish up to 2,000 to 4,000 odors. Some people can distinguish up to 10,000 different odors. So some people may have up to 1,200 olfactory receptors. Now, olfactory receptors are actually neurons. They're true neurons. They have olfactory hairs that are attached to a dendrite, a single dendrite. There's a neurosoma, or the cell body, and then it also has an axon. So the olfactory hairs and the dendrites are located in the roof of the nasal cavity. And when a chemical, when the proper chemical binds to one of these olfactory hairs, that produces a receptor potential, which, if it's large enough, it will produce an action potential in the axon of that olfactory receptor. The axons of those olfactory receptors go through the holes in the cribriform plate of the ethmoid bone, and then they synapse directly on the olfactory bulb of the frontal lobes. And so the olfactory pathways just consist of a single neuron that goes directly to the frontal cortex. And that pathway does not go into the thalamus like other sensory pathways. So this is a very, very direct route. All right, any questions? Okay, now, you may have noticed I skipped over one of the WUCLAP questions. I did change the way those are displayed on Canvas, so you should be able to see the whole questions on Canvas. So if you want to look at those, review them for the tests. Okay, so our final exam is 10 o'clock on Monday. Be sure to bring a pencil and eraser, and good luck.